Hello everyone, today we learn about disaccharides. Disaccharides, as the name suggests, are composed of two monosaccharides. So before coming to the disaccharides, we'll talk about glycosidic bond. Glycosidic bond is the one which joins two monosaccharide units in a disaccharide or a polysaccharides. So, how does this glycosidic bond forms? Glycosidic bond forms when OH of an anomeric carbon condenses with the OH of another OH group. This OH group, this alcohol group can be part of another sugar molecule or it could be any other alcohol. So, this anomeric carbon, I have already told in previous lectures that in case of aldose, C1 is the anomeric carbon, and in case of ketose, C2 is the anomeric carbon. Now, this is glucose, and glucose is an aldehyde. So, the C1, this is a C1 carbon. This OH of the anomeric carbon acts on the OH of this monosaccharide unit, and during this condensation reaction, one water molecule is released, and thus this O-glycosidic bond joining the two monosaccharide units is formed. Glycosidic N-glycosidic bond, N-glycosidic bond. It is present in nucleotides. It is present in nucleotides joining the nitrogenous base with the sugar molecule. Now, in this glycosidic bond, when this glycosidic bond is represented as, in case of malt, let's take an example of this maltose only, glucose alpha 1 4 glucose. So it means that C1 of one glucose, one molecule, is joined with the C4. So this C1 of one glucose is joined with the C4. Now the configuration of C1 involved is alpha. Here you can see this is the alpha configuration. So it is alpha 1. Alpha 1 is linked with the C4 of another glucose. This is the short representation, and this is the full extended representation. Here it is the glucopyranose form is involved, and it is the D form of the glucose. In nature, you will see that the sugars are in D form. So, and it is the alpha configuration of the anomeric carbon which is involved. So, this is alpha D glucose pyranosyl 1 4. 1 means C1 of this is linked with the C4 of D glucopyranose. Now, maltose. Uh, maltose is, if we talk about maltose, it is a disaccharide of D glucose, and specifically talking alpha D glucose. The linkage is alpha 1 4, and it is a reducing sugar. It is a reducing sugar. How come it's a reducing sugar? The C1 of this glucose, anomeric carbon, this glucose is involved in the glycosidic bond. But this anomeric carbon, the C1 of this glucose molecule, is free to act as a reducing group. So this C1 of another second glucose molecule is free. So that's why this group is potentially free, and it can reduce the cupric ion or the ferric ion present in the solution. That's how it acts as a reducing sugar. And this maltose is present in seeds and the germinating cereals. Now another sugar molecule, that is the lactose. Lactose, we all know, is naturally present in the milk. It is known as the milk sugar. It is a disaccharide of galactose and glucose. So it is the beta form of galactose and glucose is involved. The linkage is beta 1. So here one should remember that the beta is of galactose. This is the galactose molecule and this is glucose molecule. So the linkage is between the C1. The linkage is between the 1 and 4. So this C1 is of galactose and the C4 is of glucose. And it is the beta form, as you can see over here. This is this anomeric carbon, the involved in the glycosidic bond, is in beta form. So this is beta 1,4 linkage, and the anomeric carbon involved is of galactose, not glucose. So like the maltose, it is also a reducing sugar. Why it is a reducing sugar? Because here also the C1 of glucose, the anomeric carbon of glucose, is free to act as a reducing group. It is free to act as a reducing group. That's why it is a reducing sugar. Now you could come across how to learn all the structure. We have come across in my previous lecture. I have talked about in detail of how to learn all the structures. So and there I focused on, if you remember, glucose structure and learn about some facts, then you can easily draw. Now how will you draw the lactose structure? So this glucose is in pyranose form. So let's draw the pyranose form. So this is the pyranose form, HOH. This is, we are drawing glucose. Glucose, OH is over here. H, this is H, OH, H, and CH2. So we know in the pyranose form there's one oxygen and the 5 carbon. The 6 carbon is outside the ring. And here we know the glucose, it is the beta form involved. It is the beta form of glucose. So OH will be on the upper side. Now galactose. So I have told you earlier that is galactose is an aptamer of glucose. Galactose is an aptamer of glucose. So its structure will be similar like glucose, except at the C4. Glucose and galactose are C4 aptamers, which means that here the configuration differ at the C4. Configuration at galactose will be like this. In case of glucose, it is H and OH. This is H. So in case of galactose, this OH will be on the upper side. The rest of the structure will be same. CH2OH, CH2OH, OH, and it is the beta form, so it will be like this. OH will be on the upper side. Now it is the beta O glycosidic bond, so this OH will be these two OH will be involved. It is a glycosidic bond between the C1 of galactose. This is galactose, and C4 of glucose. This is glucose, so this is the C4. 1, 2, 3, and 4. So this is the C4 of glucose, and this is C1 of galactose. The glycosidic bond in lactose is between the C1 of galactose, and so this is how you will draw the structure of glycosidic bond. Okay, so now this is easy to remember, I hope. This lactose, the linkage is beta 1,4. If we talk about the extended form, it is the galactose pyranose form involved. The linkage is 1,4, and it is the beta D glucopyranose, and it is the beta D galactose. Next is the sucrose. So sucrose is made up of glucose and fructose. Glucose, we know it is an aldohexose, and fructose is a ketohexose. It is the pyranose form. This is glucose, and this is fructose. It is the pyranose form of the glucose involved, and the furanose form of the fructose. In case of glucose, C1, glucose is an aldohexose, so C1 is the anomeric carbon, and in case of fructose, C2, the keto carbon, it is the anomeric carbon. And in case of sucrose, as you can see, the C1 of glucose and C2 of sucrose both are involved in the glycosidic bond formation. That's why there is no free anomeric carbon, and that's why it is a non-reducing sugar. It contains no free anomeric carbon because both the anomeric carbon, C1 of glucose and C2 of fructose, are involved in the glycosidic bond formation. So there is no free anomeric carbon to act as a reducing group. Now the linkage is glucose. It is the alpha 1 of the glucose and beta 2 of fructose. And because both the carbon involved in this are anomeric in nature, that's why you see it is represented by a double-headed arrow. So it is a non-reducing sugar, and the non-reducing disaccharides are named as glycosides. This sucrose is present in uh, sugarcane and beetroots. Sugarcane and beetroot. It is a common table sugar we eat. It is a common table sugar, chini, that we consume. Now, it is a major form of sugar which is used in plant for transport. So in plant, the carbohydrates are transported in the form of sucrose and stored in the form of starch. Now this is known as invert sugar. Invert sugar. Why? Because the sucrose is dextrorotatory in nature. Dextrorotatory. Dextrorotatory. Which means that it can uh, rotate the plane polarized right in a clockwise direction. And when this sucrose is acted upon by the sucrase enzyme, or is hydrolyzed by the dilute HCl. Dilute It is hydrolyzed by dilute HCl or by the enzyme sucrase. This enzyme is also known as invertase. Then it will form glucose plus. Initially, it is the pyranose form of glucose. Initially, glucopyranose, glucopyranose, and the fructofurans form are formed. This is converted to fructopyranose form, which has an optical rotation of minus 92 degree. The sucrose has an optical rotation of plus 66.5 degree. The glucose has an optical rotation of plus 52.5 degree. So this is also dextrorotatory. The sucrose is also dextrorotatory, but the fructose is fructopyranose form is levorotatory, and the optical rotation of the net mixture upon hydrolysis is minus 28.2 degree. So it is levorotatory. So the sucrose is dextrorotatory in nature. Upon hydrolysis, the mixture obtained upon hydrolysis show an optical rotation of minus 28.2 degree, which is levorotatory. That's why it is called as an invert sugar, and the sucrase enzyme which hydrolyzed this is known as the invertase. Now one more sugar. This is it is a trehalose. It is a disaccharide of D-glucose, and the glycosidic linkages alpha 1, alpha 1. It means both the anomeric carbon is involved, and that's why it is represented by a double-headed arrow, because both the anomeric carbons are involved in this glycosidic bond. It is a non-reducing sugar. It is a major constituent of circulating fluid of insect, and it serves as an energy storing compound in insect. So thank you. Please subscribe my channel and share it with your friends, with your batchmate to whom it can help. Please share it with NEET aspirant, those who are preparing for CSIR net or JRF. Please do show your support so that it can motivate me to make videos further. Thank you so much.